So what are these nucleic acids? So these are the chemicals that are present inside the nucleus, which will be helpful for transmission of the inheritable characters. So have you ever wondered why we look like our parents? So why do we resemble like our father or mother or like our grandparents? Because of a molecule or because of our genes. We say genes. So I look like my father or I look like my mother. That's because of genes. But where does these genes store and how they will transmit from one generation to other generation? So where all this comes, that's on nucleic acids. Okay. So what is these nucleic acids? These are the certain chemicals that are present inside the nucleus. Okay. That will be helpful for transmission of inheritable characters like the color of the skin, the hair color, or the shape, height, everything. So whatever the physical things that you can see from a person or an organism. So all those are inheriting from one generation to other generation. And how this data or information is transmitting from one generation to other generation, that's because of a chemical called the nucleic acids. There are genes. Then how does these genes are present in the body? Where do they present? They are present inside the cell. Inside the cell, inside the nucleus, they are present. In some organisms, it is present in the form of DNA. In some organisms, it is present in the form of RNA. Most of the organism, DNA is the genetic material. The nature has selected DNA as a genetic material compared to RNA. RNA, in few viruses, it is the genetic material. So what is this DNA and RNA? It is nothing but a chemical structure, a polymer that can store all the information of an organism. So it contains all the, for suppose you take a humans contains 30,000 genes. That means they have a 30,000 proteins. So all these proteins are stored in this DNA. And that DNA, we are getting 50% from the father and 50% from the mother. So that's how we resemble like our parents. So we get half the information from the mother and half the information from the father in this chemical structure. Okay, so this DNA and RNA was present in the form of chromosomes. You might have studied in your lower classes in the form of chromosomes. And these chromosomes will be inherited from one generation to other generation. Okay, so you can say that DNA or the nucleic acids, they contain all the genetic information of an organism. So from this genetic information, the organism is made up. For suppose our life has started from single cell. That cell has divided to multiple cells and then it formed into an organism. How is it possible? Where does that information came from? It is came from the DNA. And why do we not select the RNA? Because DNA is more stable chemically, physically compared to RNA. RNA is always continuously keeps on changing. So it will keep on changing, it causes the mutations. Whereas DNA, it is chemically very stable. So this is the nature has selected the DNA. Okay, so we'll be studying about all these nucleic acids in this class. So you guys understood what I'm saying here?
Yes, sir.
Okay. So what are these nucleic acids? So the chromosomes carries the genetic material in the form of DNA, which will be very responsible for hereditary characters and genetic information. Okay. And this information is present in all the cells, not only in single cell. The same information, the 100%, the similar information is present in all the cells. Okay. So nucleic acids, there are of two types. Okay. One is the DNA and the other one is RNA. So what is the DNA and RNA? DNA is said to be deoxyribonucleic acid, whereas RNA, it is a ribonucleic acid. And what are these nucleic acids are made up of? They are made up of nucleotides. So nucleotide is a monomer, and this monomer, group of monomers come together and they make polymer. That is nothing but this nucleic acid. Okay, so just remember what is the nucleotides. These are the monomeric units. Don't confuse with nucleosides. Okay, so nucleotides are different and nucleosides are different. Okay, so nucleoside is not a monomer. nucleotide is a monomer. Okay, so this is a monomeric unit, which is nothing but a long chain of polymers, which will make the nucleic acids. I'll tell you what is the difference between the DNA and RNA. So just now you remember DNA, it is the deoxy because one oxygen will be lost here, and ribonucleic acid, it is intact, it will contain all the oxygens. Okay, so that's a basic difference between the DNA and RNA. DNA, it doesn't, it lost one oxygen, whereas RNA, it will contain all the oxygens in its structure. We'll see the structure. Okay, this nucleic acid is made up of a single unit. That single unit is called the nucleotides. Okay, it is a monomeric unit, which is a long chain of polymers. So what is this nucleotide is made up of? It is made up of a 5-carbon chain, which is a monosaccharide. So just remember, it's made up of a sugar group. It is made up of a nitrogen base, and it is made up of a phosphate ion. So just remember what is the nucleotide is made up of. It is made up of a pentose sugar, 5-carbon sugar, and it is made up of heterocyclic nitrogen. Okay, so I'll tell you why it is called heterocyclic, and it is made up of a phosphate ion. Just remember this one. So all these three together, we call it as nucleotide. DNA and RNA. So this DNA, if you see the structure, I told you that the DNA and RNA or the nucleic acids, they are made up of pentose sugar. But due to a small structural difference is causing the DNA and RNA different. So what is that? For suppose if you see the pentose sugar, it is having 5 carbon. So this is the first carbon, second carbon, third carbon, fourth, and there's a fifth carbon which is present out of the ring. Now this first carbon, it is containing the OH and hydrogen. This second carbon, it is containing the hydrogen. The third carbon, it is containing the OH. And the fifth carbon, it is containing, again, there's a OH on this. So just remember, on the first carbon, there's OH, second carbon, OH, and the fifth carbon, they also have the OH. And if you see the same structure in RNA, the first carbon will contain the OH, the second carbon will contain the OH, the third carbon will contain the OH, and the fifth carbon, which also contains the OH. I'm not drawing the full structure of all hydrogens, but these are the very important things. So just notice the first carbon, it contains OH, second also contains the hydroxyl group, third contains the hydroxyl group, and the fifth carbon also contains the hydroxyl group. Then what you can see the difference between this and this? Can someone tell? So what's the DNA and RNA as having the difference? For suppose I'll also add hydrogens here.
The second carbon has hydrogen in DNA, and in RNA it's OH.
Yeah. So whatever the ribose sugar, this is called the ribose sugar, or you can also call it as the BD ribose. I'm not going into serum chemical structures. I'm just remember it is a BD ribose. Okay, so that's a beta D ribose. And if you see here, this is BD2 deoxyribose. So why does the difference came here? On the second carbon, whatever the OH is there in the ribose sugar, that OH, the oxygen has lost here. So that's why this is called as the deoxy, as it lose the oxygen here. So that's why it is also called as BD2 deoxyribose or beta D2 deoxyribose. Okay, so that's the main difference that's making DNA and RNA. So in the RNA, the second carbon contains the OH group, hydroxyl group. In the DNA, the second carbon contains the hydrogen, not the OH. So because it is containing hydrogen, this is a very stable structure compared to RNA. Because whatever the second carbon which is containing the OH, it is very reactive group. Whereas here, it brings the stability because it is not so reactive compared to RNA. So this is structurally and chemically stable molecule compared to the RNA. So just remember, in the DNA, the second carbon lost the oxygen, so it is called as the deoxyribose sugar. Whereas here, the second carbon contains the OH group, so it is called as the ribose sugar. Understood? So why DNA is more stable compared to RNA?
Yes, sir.
Yeah. So I told that nucleotides is made up of sugar and phosphate group and nitrogen base. So this is the about the sugar. So let's see about the other things. So the bases, the nitrogen bases. So the nucleic acid in DNA, it is made up of the adenine, guanine, cytosine, and thymine. Whatever the adenine, guanine, cytosine is there, it is same present in the RNA as well. But instead of thymine, the RNA will contains the uracil. So that is another difference why the DNA is also more stable compared to RNA. So due to the thymine presence instead of uracil, the DNA is more stable compared to RNA. So see here. So RNA is made up of adenine, guanine, cytosine, and uracil. Whereas DNA, it is made up of thymine. Just remember this. So DNA, it contains adenine, guanine, cytosine, and thymine, AGCT. Whereas here, it is made up of AGCU. Instead of thymine, the RNA contains the uracil, and that is making chemically weak compared to the DNA. Next one, bases. These structures are very important. So just write down, draw. It is better if you draw. So first one is the purines. Whatever the bases that we saw here, adenine, guanine, cytosine, thymine, they were classified into two types. One is the purines and the pyrimidines. So what is purine? So just remember the purines. These are two rings. So one ring will be the six-membered, the other ring will be the five-membered ring. Okay, so both the six-membered and five-membered, they are fused together. So what are purines? These are double membrane, double ring, which is one ring is containing the six member and the other ring contains the five member. And both the six and five, they are fused. Actually, this five-membered ring is also called as the imidazole. Remember this. Whereas pyrimidines, they are containing only one ring, that is six-membered ring. They do not have the five-membered ring. Okay, so what is purines and pyrimidines? The purines contains the two rings, whereas the pyrimidines contains only one ring. Here, the two rings are one is six-membered ring, the other one is five-membered ring, both fused and make a single structure that is called the purines. So what comes under the purines? Adenine and guanine. These two comes under the purines. If you see the structure, they look like this. So if you see here, this is a six-membered, one, two, three, four, five, six. And there's a five-membered, one, two, three, four, five. And if you see the ring is made up of both the carbon and nitrogen. So these rings are also called as the heterocyclic rings. They are also said to be heterocyclic rings. So this is a adenine. This is a guanine. If you see here also there's a six-membered and five-membered ring. Whatever this five-membered ring, it is called as imidazole. And next one is the pyrimidines. So all these pyrimidines are only the single ring, which is made up of the six-membered rings. So if you see the structure, this is the structure of cytosine. This is the structure of thymine. And this is the structure of uracil. So uracil, it is present only in the RNA, whereas the DNA contains both the cytosine and thymine. And if you see here, whatever the structures, these pyrimidines, they are also present in the purines. So you can say the purines are both the pyrimidine plus imidazole. It can also be said as whatever the six-membered is nothing but the pyrimidines. And there is a five-membered, it is called imidazole. So the purines are said to be pyrimidine plus imidazole. Just remember that. So let's see how does the nucleic acids are made up of, whatever the nucleotide I said. Okay, so just we discussed about the carbohydrate, the sugar, we discussed about the base. So what are the bases are there? And we need to say about the phosphate. So all these carbohydrate, base and phosphate, they join together will make the nucleotide or nucleic acids. Okay, so they will make the monomer nucleotide, and these monomers join together, they make the nucleic acids, either the DNA or RNA. If it is a DNA, it contains the second carbon contains the hydrogen, whereas in the RNA, the second carbon contains the OH. So far, you understood? Please respond. At least comment.
Yes, sir.
What about the others?
Yes, sir.
Okay. Mandara, what happened to Nitya? She didn't join.
Don't know, sir. I did text her, but she didn't reply.
Okay. So nucleoside. So what is this nucleoside? A unit formed by the attachment of a base to the first position of the sugar is known as the nucleoside. First, suppose if you see here, so whatever the sugar, the pentose sugar is there. This pentose sugar, if it is attaches to the bases, then we can call it as the nucleoside. So nucleoside is nothing but the sugar plus the nitrogen base. Okay, so it is called as the nucleoside. And for this, if you add the phosphate group, then it becomes nucleotide. So sugar plus nitrogen base, we call it as the nucleoside. And this nucleoside with the phosphate group, we call it as the nucleotide. If it is only sugar and nitrogen base, it's a nucleoside. And you add the phosphate group, it becomes the nucleotide. And nucleotide is the monomer for the nucleic acid. Okay. And if it is OH, then it becomes the ribo sugar. If it is only the hydrogen, then it will become the deoxyribose. Just notice that one point. So based on that, the nitrogen bases can be the adenine, and if it is attaches to the sugar, then it will become the adenosine. If it is a RNA, if it is made up of ribose sugar, if it is made up of deoxyribose sugar, then it is deoxyadenosine. Guanine, it is called as guanosine if it attaches to the sugar, and deoxyguanosine for DNA. Cytosine, cytidine, deoxycytidine for DNA. Uracil, uridine, if it is a ribose sugar. And if it is deoxyribose, we call it as deoxyuridine. Thymidine, deoxythymidine. Sorry, actually for uridine, we don't have a deoxy because it only for the RNA, not for the DNA. Just notice this one point. There's no deoxyuridine because it will not attach for the DNA. It will only attach as for the RNA. Only adenosine, guanosine, cytidine, and thymidine, these are making the DNAs. Adenosine, guanosine, and uridine will make the RNA. So there will be no thymidine in RNA and there will be no uridine in DNA. Notice this one thing. So how do these things will join? So when nucleoside is linked to the phosphoric acid at the fifth position of the sugar, nitrogen base attaches on the first position. It will attaches here on the first carbon. Whereas the phosphate group, it will attach on the fifth carbon. Remember that. Okay. So on the fifth carbon, you will be having the phosphate group. And on the first carbon, you will be having the nitrogen base. So if it is only the sugar plus base, we call it as nucleoside. If it is with phosphate group as well, we call it as nucleotide. Phosphate group attaches on the fifth carbon and nitrogen base attaches on the first carbon. Notice that one thing. So nucleoside is linked to the phosphoric acid at fifth position of the sugar and it gets the nucleotide. So it will happen like this. So here there will be base will attach here. It can be the hydrogen or OH based on DNA or RNA. If it is DNA, it is H. If it is RNA, it will be OH. And there will be OH here. And on the fifth carbon, there will be OH. And if you see the phosphate group is having OH, 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 and double bond O. So this is normal structure. But if it becomes an ion, it will lose all the hydrogens and turn out to be O minus, O minus, O minus. So when it loses the hydrogen, and on the fifth carbon, whatever the OH is there, that OH also will break and they will remove like a water molecule. And this O directly attaches to the fifth carbon. And that's how the bond will happen here. So this OH will, whatever this oxygen is there, it will come and attach here. So all are containing negative charges because they are losing the hydrogens and they are turning out to be ion. So because of this phosphate group, the DNA is said to be negatively charged. Remember this. So whatever the OHs are there on the phosphate group, they will lose the hydrogen and they will turn out to be O minus. Due to that, it will give the negative charge for the DNA. So if a question comes like DNA is positive charge or negative charge or neutral, you should say it's a negatively charged because of the phosphate group. The phosphate groups are containing OH, they lose the hydrogen and they turn out to be O minus. And one such O minus, here it loses the hydrogen and the carbon loses the OH on it and they leave like a water molecule. So this bond is called as the phosphoester bond. Remember. It's a phosphoester bond. And whatever the bond is present here, this one is called N-glycosidic bond. Nitrogen base with a sugar. So it's called as N-glycosidic bond. So you can see here. So there's a fifth carbon that is having OH and on the phosphate there is a OH. So both will leave like a water molecule and they will turn out to be water. They will lose like a water molecule and there will be bond formation happen here. So if it is only one phosphate group, then we call it as monophosphate. And if you see here it didn't use the word nucleotide because sugar plus base, it is a nucleoside and that is attaching to one phosphate. So nucleoside monophosphate. We should not use nucleotide monophosphate because we are separating this and this. So we are using nucleoside monophosphate. And if it is the other phosphate, which is also containing OH, double bond O, OH, and there is a OH. And there's this phosphate, which contains the OH, OH, double bond O. There's a OH, which will attach to the carbon. So now this H and this OH, they will lose like a water molecule and they will join here. So they will directly join here. So by losing the water molecule and the joining here, so this bond, so you can say there's a condensation happened here. Okay. Just remember here. So by losing the water molecule, the bond has formed here. But this is not a it's not called the ester bond because these are not the esters. Okay. So just remember this. So here the dehydration has happened. Water has lost here. And they made the bond here. And the same happens with this phosphate as well as this phosphate. So whatever this phosphate is there, it is containing the OH. So they will lose the bond and they will join together. Okay. So there will be condensation happened here by losing the water molecule. So if it is only one phosphate, we call it as the nucleoside monophosphate. If it is two phosphate groups, it is a diphosphate. And if it is three phosphate groups, then it is called triphosphate. And remember, whatever the bond present between the oxygen and phosphate, they are containing a lot of energy. So when you break down this bond, it will liberate a lot of energy. That's why we called ATP as cellular currency. If you might have studied in lower classes, we say that ATP is called the cellular currency. Why? Because they contain so much of energy between these phosphate bonds. And if you break down this phosphate, you will liberate a lump of energy. So ATP when convert into ADP, ADP when convert into AMP, they releases a lot of energy. So whatever this ATP is called as the cellular currency because of this. It holds the energy between the phosphate groups. Understood? And whatever the base and first carbon, they are attached with the glycosidic bond. We can also call it as N-glycosidic linkage. That's happening between the nitrogen and the sugar. So we call it as N-glycosidic linkage, or you can simply say glycosidic bond. Understood? So how does the structure has formed here? So how the phosphate group is attached to the fifth carbon, and how one phosphate group is attached to other phosphate. So how does the two nucleotides will join together? First, suppose I told you there's a pentose sugar, which is having the OH. It could be H or OH, depends on whether it's a DNA or RNA, whether it's ribose sugar or deoxyribose sugar. And there's a base will attach here. And there is a fifth carbon. This fifth carbon will attach to the phosphate group. You can write like OH or you can put O minus. It depends on you. Okay. And whatever, if you see the third carbon, it is containing the OH group for both the DNA and RNA. And if you see the fifth carbon also do have the OH. So see here, let's draw another one. So let's say it's a DNA. So I'll put hydrogen here. So there's OH and there's a carbon which is having the phosphate group. The phosphate group is having OH groups. And there's a base. Now see this one. So the phosphate group is having OH group on the fifth carbon, and on the third carbon there is a OH group. Again, the same way happens here. So there is a loose of hydrogen and OH will make water molecule, and they will join together with the help of this oxygen. So if you see here, there is a phospho-ester bond in this, and there is a phospho-ester bond in this. So both together join. So we call this as the phosphodiester bond. Two esters were joining together. The bond between the fifth carbon and phosphate group, this is called as the phosphoester bond. And whatever the bond between this nucleotide and this nucleotide, it happened by losing of the water molecule. So this is a phosphodiester bond. We'll not say here decondensation. We'll only say between the phosphate and the other phosphate, that bond is called as the anhydride bond because it the bond is formed by losing the water molecule. Okay, so this is called as the anhydride bond by losing the water molecule between the two inorganic compounds. So this phosphate and there's another phosphate. Whereas if you see here, this is one ester and this is one ester. So it happened between the two esters. So we call it as a phosphodiester bond. Understood? So the phosphodiester linkage has happened. And if you see, this is the fifth carbon and this end is called the third carbon. So this is fifth end and this is the third end. So if you see the linkage or the phosphodiester bond is happening between the fifth and third carbon. So see here. So the phosphodiester linkage will happen between the fifth and third carbon of the pentose sugar because this one is third end and this one is fifth end. On the fifth end, you have this phosphate group, which is having the OH, and third end, there is a OH. By losing the water molecule, there is a joining was happening. And it's happening between the two esters, so we call it as the phosphodiester linkage. And here the water molecule is lost. That's why this is called anhydride. And this is called an glycosidic linkage, or you can say simply glycosidic bond. Is it clear? So how the two nucleotides were attached? You can see here clearly. So this is the fifth carbon that is having the phosphate. And you see here, this is the fifth carbon that is having phosphate. And this phosphate and the third carbon, they join together. So it happened between the third carbon and the fifth carbon phosphate. And there are nitrogen bases which are present here. So that's how a group of nucleotides will join together. And this is a DNA because there is a H here. If it is having OH, then we can call it as RNA. So you can see. So there's a sugar. This sugar is having on the first carbon, the nitrogen base. On the fifth carbon, the phosphate group. This phosphate is attached to the third carbon of the pentose sugar. and that will keep on repeating, and this is a monomer. And this monomer will make a polymer called the nucleic acid. It can be the DNA or it can be the RNA. So next one. So who has given this DNA? So this is how the DNA or nucleotides, group of nucleotides will join together. And if you see this base will connect to the another base of the another nucleotide of the opposite strand. If you see, the DNA means you think like this, the helical structure. This is what the DNA you see. So this is one strand. That one strand and the other strand, they join together by these hydrogen bonds. So you see here, this is a one chain and this is the opposite chain which is running in the opposite direction. So we call them as the anti-parallel strands. And both are joined between the nitrogen bases. So we call it as the hydrogen bond, which is joining two bases of the opposite strands. So that structure is given by the James Watson and Francis Crick, who has given this double strand helix structure. So there are two nucleic acid chains which are wound about each other and held together by the hydrogen bonds between the nitrogen base pairs. So how does that happen? So suppose you see here, there is a pentose sugar. Here there is a phosphate group is present. Here there is a phosphate group is present. And if you see on the first carbon, there is a nitrogen base is present. This is a single ring and this is a double ring. So what are single rings are called? Can someone tell me? What are single rings are called? Purines or pyrimidines?
Pyramids.
Single rings are called, yeah, they are called as the pyrimidines. And whatever the double ring is there, it is called as the purines. Okay. So just remember one point here. Always a purine will join with a pyrimidine with the help of hydrogen bond. This one thing you need to remember. Purine joins with the pyrimidine with the help of the hydrogen bond. For suppose you see here, thymine. Thymine is a pyrimidine that joins with the adenine, which is a purine. Okay. And you see here, cytosine. Cytosine is a pyrimidine that joins with a guanine, which is a purine. So just remember the purines and there is a pyrimidines. The purines are adenine and guanine, whereas the pyrimidines are thymine and cytosine. So can adenine join with cytosine? No. There is a complementary base pairs we say. That means adenine can only bond with thymine. Guanine can only bind with cytosine. So see here, adenine always binds with thymine with the help of a hydrogen bond, and guanine will bind with cytosine with the help of hydrogen bond. Guanine cannot bind with thymine, adenine cannot bind with cytosine. So always the linkage will be like this, ATGC. So it's a adenine and thymine, guanine and cytosine. So between adenine and thymine, there are two hydrogen bonds are present. Between guanine and cytosine, there are three hydrogen bonds are present. You can see here. So this is one hydrogen bond. There's another hydrogen bond between the thymine and adenine. And there's a three hydrogen bonds between the cytosine and guanine. If in case of RNA, adenine binds with uracil with a double bond, guanine binds with cytosine with the help of triple bond. So that's the difference. So this is for DNA and this is for RNA. So in place of thymine, RNA will contain the uracils. But guanine, cytosine, adenine are same between the DNA and RNA. So it's a common between the DNA and RNA. So you need to notice few things. So between the thymine and adenine, the nitrogen bases are connected with the hydrogen bonds, and these are complementary. That means adenine can only bind with thymine. It cannot bind with the cytosine. And the same way, guanine can only bind with cytosine. Adenine can bind with uracil if it is a RNA. Guanine can bind with cytosine. So that's how they will be making the complementary base pairs. So we call it as complementary base pairs. So remember this one thing. And always remember the purine will join with the pyrimidine with the help of hydrogen bonds. RNA. So if you see the RNA, these are only single stranded. They cannot make the double strand like a DNA. So this is a single stranded. Whereas DNA, they make the double stranded, the helical structure. But RNA can only make a single strand. But sometimes what happens if there is a complementary base pair, for suppose adenine, and there is some uracil was present here. So this can fold and they can join like this. So if there is any complementary base pairs are present, they can fold and they can make a double helix structure. So see here, RNA are only single stranded, but sometimes they fold back on themselves to form a double helix structure when there is a complementary base pairs were present. RNA are of three types. One is mRNA, rRNA, and there is a tRNA. It is mentioned in the NCERT in chemistry textbook. So I'm telling this. So what is this mRNA? I told you the genes were stored. The nature has selected the DNA as its genetic material. But when they are synthesizing the proteins, they cannot make it directly. So whatever the so DNA is like a database. So it's a data center where all the data of the cell or organism is present, and it is well protected. This database cannot directly give to the output. So there should be someone who intermediate between the DNA and RNA. So that is between the DNA and proteins, the intermediate person is the RNA. So DNA will give a piece of genetic information and the RNA will take the genetic information and it will make the proteins. So this is called as the central dogma. So the so whatever the information is there, all the information, it cannot pass at one time. It can only pass one kind of RNA. So the DNA will just give only a piece of information, which can only make a single protein. So that piece of information is nothing but the RNA who will make the proteins. So there is a DNA and proteins. The one who transfers the genetic information is nothing but the mRNA. So that's why this guy name is called the messenger, because he read the DNA and he will make a piece of genetic information that's called mRNA, and he will go and he will synthesize the protein. But how does that happen? So this mRNA, when it comes to ribosomes, you might have studied in class 9th or 10th. So these ribosomes, which contains this RNA. So what does they do? They will read this guy. They will read this guy and they will tell the tRNA to bring particular protein, particular amino acid. For suppose, let's say the DNA is like ATGCAA. So it's a double stranded. So A is complementary to T, T is A, G is C. So this is how there are lakhs, millions of nucleotides which are having a particular sequence. Now this guy will give information just only the complementary. So it will be like U, because instead of T, you will be having uracil. So uracil, and there will be A, and there will be C and G. So this is messenger RNA. So you will come out of the nucleus and you will go to the ribosomes where this RNA is present. You will start reading this code. So let's say this is a code. You will start reading a series of code. So every three base pairs, from every three base pairs, he will make a one amino acid. So this guy will read a three nucleotides and he will tell the tRNA to bring that particular amino acid. He will read these three nucleotides and he will tell the tRNA to bring that particular amino acid. So all these amino acids will join, and it will make a chain. So that chain is called the amino acid, nothing but a protein or peptide chain. You might have studied the proteins. So understood how the information has passed from DNA to the proteins. Just remember, DNA cannot synthesize the protein. For that, we need an intermediate molecule that's called mRNA. So this DNA will just give a piece of chunk of information. So that information you will carry to that rRNA. That rRNA will read this information. For every three nucleotides, it will code for one amino acid. It will tell this information to tRNA to bring particular amino acid. So those amino acids will be brought by the tRNA and they will be arranged in a series, and that will make a protein chain. So that's how there are different types of RNAs and how they will be helpful. Understood, guys?
Yes, sir.
Okay. So only Gauri understood. What about the others?
Yes, sir.
Okay. So next one, DNA fingerprinting. So what is it? So if you see any person, so from one person to other person, 99% of genetic information or 99.99% genetic information is same. But there is a 0.1% or 0.1% which is different. So that difference will be helpful for identifying who is the suspect in a crime scene. For suppose there's a crime happened. In that crime, I got a hair follicle. Okay, I got a just a strand of hair. So from that hair, I'll extract the DNA. And so those who involved in this crime, I know there are few suspects. Let's say the suspect A, suspect B, suspect C. So who are present at the time of crime scene. So what I'll do, I'll extract the hair from the suspect A, suspect B, and suspect C. So from these guys, I'll take the DNA and I'll compare the DNA with this. So 99.99 is same, but that 0.1% or 0.01%, the difference will be able to identify whether it's a suspect A or suspect B or suspect C who is criminal. So that's called the DNA fingerprinting. So what is this DNA sequence? It is nothing but the sequence of DNAs, like it's nothing but A, T, G, C, A, A. So there will be code like this, a series of code. Some millions of nucleotides will arrange in that sequence. So that will be unique for every person. So 99.99 is same, but that 1% or 0.1% is different. That will be helpful for identification. So that's called the DNA fingerprinting. So how do we use this DNA fingerprinting? Where do we use? One thing will be used in the forensic. So for identifying the criminals. Second, we'll be using for to identify who is the father or who is the mother. So there's a identifying for the paternity. Sometimes there's a dead body. We cannot tell whose dead body is that because the body is completely decayed or the body is completely burnt or something. We cannot identify. So at that time, for identifying the dead body, we can use the DNA fingerprinting technique. So these are the three applications where you will be using the DNA fingerprinting. So what we'll do, we'll collect the crime scene sample and we'll analyze the crime scene sample. So we'll identify that and we'll find out who is the suspect by comparing the DNA between the crime scene and the suspects. So that's about the nucleic acids. And there's a small part of hormones is there. So what are hormones? These are nothing but the intracellular messenger. For suppose in our body, there are two ways the message will be transmitted. One is with the help of neurons, nerves. The other is through the chemical substances. So those chemical substances are nothing but the hormones. So what are hormones? These are nothing but the chemicals which are secreted by glands, the endocrine glands. So remember, if any options they mention like exocrine, just notice it's endocrine, which do not have any duct. That means they will be directly released into the blood. So they will not be having any tubes that will carry to the target place. For suppose hormone is synthesized and secreted in one place, but the action of this hormone will be happening at another place. So this is the target place. So if it is carrying through the closed duct, closed pipe, then we call them as exocrine glands. But if it is just released into the blood and wherever the blood goes, the hormone is carried away, then we call them as the endocrine glands. Okay, so hormones are secreted only by the endocrine glands, not by the exocrine glands. Remember this. Okay, so which are ductless glands. So they will be released directly into the blood. So just remember, so these are the products. These are produced by the endocrine glands in the body and are poured directly into the bloodstream, and the blood perfuses to different places. So it also carries to different places. So the site of action, or you can say the target place. And this hormone will act on that target place. So what are the different kinds of hormones are there? So they are classified chemically: steroids, polypeptides, and amino acid derivatives. So what are steroids? They are nothing but the carbohydrates. Okay. So polypeptides, which are nothing but the proteins, and the amino acid, they are also uh, amino acid. They are also related to the proteins. So steroids, estrogen and androgen. That's a male and female hormones. So polypeptides, which are insulin and endorphins, epinephrine and norepinephrine. We'll see what are they. So what does the hormones will do? They will take us several functions in the body. They help to maintain the biological activities. For suppose, let's say you have eaten the food. So what happened? The food will digest into simple glucose. This glucose will be absorbed into the blood. So when it immediately absorbed into the blood, what happens? Your blood glucose level rises. So usually it should be 110 or less than 110, or you can say 70 to 110. But after you eating the food, the glucose will just spike a lot. In order to decrease that glucose and maintain the optimum level, the insulin hormone will release. So what does the insulin hormone will do? It will just open the cells, some kind of channels. So when the cells open those channels, the glucose will enter inside the cell. So whatever the glucose that is revolving in the blood, it will just pass into the cells. So the blood glucose levels decreases. So that's how the insulin will limit the blood in the limit the glucose in the blood. Then what does a glucagon will do? For suppose you are not eating any food, so you are fasting, so there will be no glucose, so the blood glucose will fall. So whatever the glucose that is stored in your liver or is stored in your adipose tissue, so that will be released back into the blood. So that's how the low blood sugar will increase with the help of glucagon. So they will be altering based on your food diet. So that's what insulin and glucagon together regulate the glucose levels in the blood. So when you are doing the long fasting or intermediate fasting, there will be more glucagon in your body that will help to reduce the body fat. When you are eating too much very frequently, you are spiking always the insulin. So what it will do, it will accumulate all the glucose in the cells and it will helpful for increasing the body weight. So that's why the intermediate fastings will be helpful for decreasing the body fat. Next one, there is the other hormone called the epinephrine and norepinephrine. For suppose when you are threatened, at that time this epinephrine hormone will be released. So what does it will do? It will just make you alert in the situation. So suppose whatever the goosebumps or dilated your eyes to see better in the dark. So all these because of this epinephrine. Okay, so that's a fight or flight hormone. So that will be helpful for making you alert. And norepinephrine, for suppose when you're sleeping, your body is very rest and it is so peaceful. At the time, your body doesn't need to be so alert. At the time, this hormone will be secreted. So that's a help of the norepinephrine. So which will mediate response to the external stimulus. So based on what you see, what you think, all that will helpful for either epinephrine or norepinephrine. For suppose you are so thinking about something uh, and occupied with this, and you got so much of anger or fear, that will lead to production of the epinephrine. So all these because of the external stimuli. And there's a growth hormone and sex hormones, which will be helpful for increasing your growth and helpful for increasing your sex organs. There's other hormone called the thyroid hormone, which is produced by the thyroxine, which is a amino acid. So this thyroxine will join with the iodine to make the tyrosine. Okay, so just remember thyroxine is an iodinated derivative of amino acid, which will be helpful for thyroid hormones. So whatever the T3 and T4 hormones, thyroid hormones, it's made up of this. So if this thyroxine is very low, abnormally low, then the person is said to be hypothyroidism. So the person will be having low metabolic rate. So when the person is having low metabolic rate, whatever the food that he eats, it will not be metabolized, it will not be utilized, so it will store. So the person will become so fat when the person is suffering with hypothyroidism. And in the increased levels, that will cause the hyperthyroidism. So that will be showing some other symptoms, like the person will be overactive, all those. So mostly people will suffer with the hypothyroidism. That's a less amount of thyroxine. And we know this thyroxine is just made up of iodine. And if you are taking iodine very less, so that will cause low secretion or low synthesis of thyroid hormone. So that will cause the enlargement of the thyroidism. Okay. And that leads to very lethargy because your body is so weak as it is not metabolizing a lot. So decreased metabolic rate will make you obese and it will make you lethargy. That means very weak. So lack of energy, always tired. So that's a hypothyroidism. So how do we control that? The government has initiated, for suppose whatever the salt we are using, they made that every salt should contain this iodine. So no population will suffer with lack of thyroid hormone. So that's how the government has tackled with the help of iodized salt. And steroid hormones, it's a adrenal cortex and gonads. So glucocorticoids, mineralocorticoids, which are all secreted by the adrenal cortex. This is the area in your brain which will be helpful for producing these hormones. So what does the glucocorticoids will do? It will be helpful for metabolism and whatever the inflammation you're seeing, like allergy, so the cold and cough, all this because of this glucocorticoids. It will be helpful for dilating your blood vessels and reduce the allergy reactions. So it also try to reduce your stress. So suppose when the person is suffering, is having too much of stress, the cortical hormone will be released a lot. That will try to reduce the stress. So that's how the doctors will test if the person is having too much stress or not by checking out this hormone. So next one, mineralocorticoids control the level of excretion in water. For suppose the person is having too much of water, what happened? It will put load on the heart, and that will increase the blood pressure. So the person will affect with the heart. So we need to reduce the water. For suppose you are drinking only 4 liters, that's okay, fine. But if you are drinking too much, like 8 liters or 10 liters of water, what happened? There will be so much of accumulation of water in your body that will directly affect your blood because your blood levels also increase. That will put the pressure on the pump. And what is a pump? Nothing but the heart. So our body try to eliminate that excess amount of water. That happens because of these mineralocorticoids. If you are drinking too much of less water, you are dehydrated a lot. Your body wants to conserve the water. That will happen by the mineralocorticoids. So which acts on the kidney, whether you need to send out the water more through the urine or you need to conserve the water in the urine. So this hormone will decide. Next one, testosterone. You might have known this, which will be helpful for secondary male sex organs for development of their so deep voice, facial hair, general physical constitution, so all these. And estroidal, that's the estrogen in the females for developing the secondary females sexual characters. And the progesterone, this is a very important because it will be helpful for maintaining the pregnancy. Okay, so whatever the after fertilization, whatever the fetal that is developing in the mother womb or in the uterus, so that will be well developed only when you are having enough amount of progesterone. If you are having lack of this progesterone, that can detain the baby's growth. Okay, so for that, progesterone hormone is very needed. Sometimes the doctor will give externally by making these hormones.